Hello everyone, I'm Dr. Tanushree and I welcome you all to the question paper discussion session of CSIR Net exam held this year on 17 September. Today in this session, we are going to discuss questions of Unit 13, that is methods in biology. This time, total 10 questions were asked from Unit 13. And the interesting fact is that the questions were framed from topics from almost all the subunits of Unit 13, like subunit A, C, D, E, F, G, and H. Questions were framed by combining the concepts from other units as well, like from Unit 13 and Unit 3, 8, and 12. Statement based, figure-based, numerical type, and match the following type questions were asked in the exam. So let's have a look at the question. The very first question is from subunit 13D, that is statistical methods. The question is about what is the 50th percentile of the numbers 9, 5, 11, 3, and 2. Now, you can only answer this question if you know what is 50th percentile. 50th percentile is the value where 50% of the data fall below it and 50% fall above it. So this is same as definition of median. Same as definition of median, which you study in subunit 13, these statistical methods. So options given are 5, 6, 9, and 15. So question arises how to calculate 50th percentile. So for this, we have to follow certain steps. The very first step is that whatever values, whatever numbers are given to you in the question, you have to order them from smallest to largest. So 2, 3, 5, 9, and 11. This is how you're going to arrange them. Then, because as I mentioned that 50th percentile is the value where 50% of data fall below it and 50% fall above it. So therefore, we will be multiplying 50th percent by total number of values. So total number of values are 5, right? So this is the reason we are simply multiplying 50th percent by 5. So this gives you 2.5. Now the next step is that you have to, like, if it is not a whole number, like in this case we are getting 2.5, so it is not a whole number, so we have to round it up to the nearest whole number, which gives 3. This means that in the arrangement of the values that we have done, we have to look which number comes at the third position. So that means it is the number 5. So when we count the values in our data set from left to right, that is from smallest to largest value, the number which is indicated in step 3, that is values up to 3, we have to count. So that is our answer. So the corresponding value in our data set for the 50th percentile is 5. So therefore, answer is option 1 here. Moving on to another question. Now see, this question is from subunit 13H, that is methods in field biology. And it's a very direct memory based question. The question is about remote sensing. Like in remote sensing, which one of the following formula is used for the calculation of NDVI, that is normalized difference vegetation index? So here you have to simply recall the formula for NDVI. So it's a very direct question, memory based question, and here the correct formula is that NDVI, it is the difference between near infrared and red reflectance divided by their sum. So therefore, answer here is option 4. Moving on to another question. Now this question is from subunit 13E, that is radio labeling techniques. The question, I'm sure most of you have done it correctly because the question was very simple. If you have understood the concept, if you have understood the question thoroughly, you must have answered this question particularly correctly. So the question is that radioactive isotope of an element has a half-life of 100 hours. How many hours will it take for 15 by 16 of the source amount to decay? Or in another way, I can say how many hours will it take for 1/16 of the source amount to remain. So if the initial source amount is 1, definitely after one half life, it will be reduced to half. Again, after another half life, it will be reduced to half. And again, it will be reduced to half, and so on. So we can see that when the 5 lives have passed, the source amount remaining is 1/16. And one half life is of 100 hours, right? So we have a half life of 100 hours. So that means if I write 100 here, then again 100, 400. So let's see if the 400 is given in the option. So yes, in option 2, it is given. And therefore, it is the answer. So this is the explanation of the same that I explained in the, like, this is the theory of the same, the text of the same that I explained in the previous slide. Moving on to next question. Now, this question is from subunit 13C, that is biophysical methods. This question is on spectroscopy. Now, if you have attended the classes, I always say in my class that whenever you are studying subunit 13C, when you are preparing spectroscopy, you have to prepare all the spectroscopic techniques, because when the combination based, like they give you a statement and in the options they give you all the spectroscopy technique, such type of question you can only answer if you know about all the spectroscopic techniques, right? So this is that kind of question. So you can see it on your screen. So these are the options given. So this particular question is about spectroscopic experiment that would help distinguish between alpha helix, 3 by 10 helix, or pi helix. So that means here you have to find out which spectroscopic technique is used for studying secondary structures of protein. So in the options, it is UV given, UV spectroscopy, fluorescence, NMR, and circular dichroism. I'm sure most of you must have been get confused between option 3 and 4, because we all know UV is an absorption spectroscopy, right? So we use UV visible spectroscopy for qualitative and quantitative analysis of a sample, right, for determining concentration of the proteins and other biomolecules. Fluorescence is an emission spectroscopy, right? So basically, fluorescence spectroscopy, you can use it to study the structural changes in conjugated systems or aromatic molecules, rigid, planar compounds, and even to study the interaction between one protein with another, receptor ligand interaction, right? So when it comes to NMR and circular dichroism, yes, both of them are being used to study the structure, secondary structure of the proteins. But the point is, the one word, if you have seen here, near UV circular dichroism. So if you recall which UV region is used for studying the secondary structure, based on that, you can answer this question. So it's not the near UV, it's the far UV that is being used. And therefore, option 4 cannot be the answer. So you're only left with option 3, that is NMR spectroscopy, and that is your answer. So you can see such minute details, like when you're preparing for CSIR exams, such minute details you have to keep in mind, right? So even if you have done this question wrong for the, like, just make sure that like you have to, like, when preparing for CSIR exam, these fine details has to be kept in mind. That is what the point I want to say here. So far UV region of the light is used for studying the secondary structure of proteins and not the near. Now, coming to next question, if you see this question is based by combining concepts from unit 3 and unit 30. Basically, in unit 3, you study about recombination, right? You study about RNA interference. And even in homologous recombination, you study in unit 8 also. So basically, the question is that a table is given to you, and here in column X, they have given different ways of carrying out reverse genetics. So techniques they have given for carrying out reverse genetics and strategies to achieve the same. So which one of the following options is a correct match between X and Y? And these are the options. So basically, when I use a term random mutagenesis where we want a mutation, like a random mutation is required, right? So that means random, not targeted. So that means we need something which can induce mutations randomly. And for that, we all know transposable elements are the ones which generate random mutations. And even uh, like chemical agents or physical agents like UV rays. UV rays can be used. So that means A, it matches with 2 and 4. So in the options, we have to see where such option, such combination is given. So if we see option 2 and 4, such combination is given. A2 and 4, A2 and 4. But now to confirm the answer, like obviously we have omitted 2 options out of 4. But now what is the correct answer? 2 or 4? So for that, we have to see the B also, that is targeted mutagenesis. Now here you can see a term targeted. So that means here, if we want a targeted mutation to happen, for that the best strategy is homologous recombination, and even the CRISPR, which is a powerful gene editing tool that we use, the scientists, the researchers use nowadays. So B should be 3 and 5. So if you compare option 2 and 4, this combination is given in option 2 only, and therefore this is the correct answer. And the phenocopying that now we can easily make out, that is RNA interference. So that means you have to be sure about two things, right? Like one, to omit 2 options out of 4, and another to confirm the answer. So you need to be here, you need to be thorough with either phenocopying or random mutagenesis or targeted mutagenesis. So here the answer is option 2. Coming to another question. Now you can see this is the another question from subunit 13D, statistical methods. If the weights of 10,000 seeds from 100 individuals of tree species are measured, which one of the following distributions is expected? So in the option, it is binomial, Poisson, Gaussian, and no predictable distribution. So basically, if you go through this question, you can easily answer this question if you have studied about these statistical methods, these probability distribution methods in detail. So basically, when we talk about binomial, right? So by prefix means two. So binomial distribution is applied only to those samples that have only two outcomes, right? Like success or failure of an success or failure outcome in an event in an experiment or a survey. Like for example, when you toss a coin, you'll get either head or a tail, right? However, when we talk about Poisson distribution, it's its samples can take only integer values, right? And integer values are basically, they are whole numbers. They can have positive, negative, or zero value, but cannot, the values cannot be in fraction or decimal. So when we talk about here the seed weight, right? So seed weight can be in decimal, like the seed weight can be 7.5 gram, right? So that means Poisson distribution will not be the answer. Then when we talk about Gaussian distribution, so yes, it will be applicable here because it's a continuous distribution and it can take any value. So that means to answer this question, you should know what is binomial, what is Poisson, and what is Gaussian distribution. And therefore, the answer here is option 3. Now, moving on to another question. Protein A was subjected to different experiments. First, SDS page with or without beta mercaptoethanol. Second, fluorescence. Third, far UV. And fourth, near UV-CD spectra at pH 7 and 3 red. So the results are shown below. So basically, these are the results shown, right? First, second, and this is the third, and this is the fourth. So for STS page, for fluorescence spectra, for far UV, and for near UV CD. And these are the options provided. And it has been asked that which one of the following options provided the correct inference. So that means to answer this question, we have to read the options we have to go through the options one by one, right? So the first option is that protein A is an SS bonded homotetramer and each subunit has a molecular mass of 50 kilodalton. So first you need to recall here what is the function of SDS, sodium dodecyl sulfate. So sodium dodecyl sulfate, that is SDS, it's an anionic detergent and it is needed for denaturation of the proteins, right? And it unfolds the protein structure, right? Now in this first figure, with and without mercaptoethanol, we have to check. Now, what is the role of beta mercaptoethanol? It's a reducing agent and it is responsible for breaking the disulfide bridges. So here you can see that when the beta mercaptoethanol is not used, we are getting one single band of 200 kilodalton. However, when we are using the beta mercaptoethanol, we are getting a single band of 50 kilodalton. So this indicates that the that on when we are using beta mercaptoethanol, it breaks the disulfide bridges. So that means our protein comprises of 4 subunits, each of 50 kilodalton, because that only totals 200 kilodalton, right? So yes, it is right that protein A is a homotetramer made up of 4 identical subunits and constituting a final weight of 200 kilodalton. Now, the second part of the option 1 says that folded at pH 7 and molten globule at pH 3. So each subunit has a molecular mass of 50 kilodalton, folded at pH 7 and molten globule at pH 3. So where this pH data is given in figure 4, that is near UV circular dichroism. Now the blue one represents at pH 7 and the red one represents at pH 3. So basically you can see there is an increase and decrease, right? Increase and decrease in the like, you can see this curve, but the red one is constant, right? So this indicates that our protein is folding, right? And that because that is the reason different wavelengths we can see here. But because at like already when it is in folded state, which has been shown by this red line, so therefore it is a constant value. And therefore, this confirms our answer, and therefore answer is option 1. But always it's better to read, like it's always better to confirm by going through all other options. So just by going through option 1, we can easily make out that this is the answer. But yes, we should also go through 2, 3, and 4 options. So in option 2, it is mentioned that protein A has a molecular mass of 200 kilodalton. Beta mercaptoethanol degrade the protein. No, this is wrong. Beta mercaptoethanol doesn't degrade the protein, right? It breaks the disulfide bonds. Then third option is SDS denatures protein A into different oligomeric state. No, here only one oligomeric state is there. So therefore, this cannot be the answer. Then option 4, SDS promotes the formation of different oligomeric states. So again, not different, only one. So again, this cannot be the answer. So this confirms our answer. Answer is option 1 here, right? Now moving on to another question. This question is from recombinant DNA technology, restriction enzyme from subunit 13A. Given below are names and recognition sequences of few restriction enzymes that are used for cloning experiments. So you can see here, these are the restriction enzymes which have been mentioned, and the cleavage site of each enzyme is indicated by this asterisk sign. Now, given below are different vectors, right, and insert. So vectors are given in column A and column B fragments, like vectors and insert fragments are given. Vector fragment in column A and insert fragment in column B, generated by digestion using the above enzyme, that is these enzymes. Now, which one of the following options represent correct combinations of vector and insert respectively, which generate compatible ends for ligation? So here in this question, you have to recall the concept of blunt end and sticky end ligation, right? So these are the options given. So let's find out which vector fragment will be compatible with the insert fragment. So the one thing is very much clear, that in the recombinant DNA technology, when you're constructing a recombinant DNA molecule, you ligate a vector with a fragment, DNA fragment carrying your gene of interest. So ligation is done by ligase enzyme. But basically, this the perfect like the binding of the vector with the insert will take place if they have the sticky ends, compatible ends they have, right? So to use vectors and insert fragments for cloning, they should have compatible ends. So that means here we have to compare, right? So if you see all the restriction enzymes, we have two enzymes which generate the blunt ends, right? So basically, what are the enzymes which generate the blunt ends? So here you can see EcoR5 and we have HINK2. So you can see here, they generate blunt ends, right? Because here the dilation is just from the center of the recognition sequence, right? So they don't generate sticky ends, they generate blunt ends. However, if you see EcoR1, BAMH1 and BG3, they generates the sticky end. So you very well know that when we talk about the blunt end, right? In case of blunt end cutting, any end could be compatible with any end. So this means that EcoR5 will be compatible with HINK2, right? Or here HINK2 and EcoR5. So that means we have B2 and E1. So these combinations can be used in cloning, right? So this is about blunt end, like when the enzymes generate blunt end. What about when the enzyme is generating sticky ends? So when the enzyme is generating sticky ends, like we know that this insert fragment will bind to the fragment, vector fragment. So these should be compatible, right? Like if A is present here, it should be T here. If G is present here, C should be here, right? A should be present here. So they should be complementary. So again, here we have to compare the fragments, right? So the sequences are already given to us. So basically, if I compare BG3 with BAMH1, so basically the sequence given to us is of BG3 is A, G, A, T, C, T. So this is 5' to 3'. So I can write down the complementary sequence. So it will be A, G, A, T, and C, right? So if I compare it with BAMH1, that is G, G, A, T, C, C. So this is again 5' to 3'. That is G, A, G, G, A, T, C, C. This is 3'. So question arises, which sequence will be compatible, right? So if I compare these two strands, right? So this one is compatible, this one is compatible, this one is compatible, and this one is compatible, right? So this means that these two combination, that is D4 combination, can be used, because here we are like on using these two, we can generate a compatible ends. Like these two enzymes can generate the compatible ends that can be used for cloning. Right? So answer here will be option 2, because we should get this combination. So if you go through the options, this option is given over here in option 2 only, right? Now moving on to another question. Now this question is from subunit 13F, microscopic techniques. This question is very direct, like memory based. Previous question that we have seen, it required conceptual clarity. But this question simply required it's simply a memory based question, and you have to just simply recall about the principle and application of microscopy techniques. So here the two microscopy techniques which are given are two photon microscopy and confocal microscopy. So if you have studied about these two, you can easily answer this question, right? So it's a statement based question. So let's see. what all information is given in the question. So here the properties of two photon microscopy over traditional confocal microscopy is given, and four statements are given, and we have to find out which of the following combination of statement is correct. So if you compare statement A and B, they are contradictory. In A statement, it is mentioned by using longer wavelength, two photon microscopy induces less photo bleaching of the tissue preparation, or by using shorter wavelength. See, only differences in only one word here in both these two statements. So by using shorter wavelength, the two photon microscopy induce less photo bleaching of the tissue preparation. So this you can only answer if you have studied about these two microscopy techniques. So when we talk about two photon microscopy, it can penetrate deeper into the tissue than confocal, because the excitation wavelength which is being used here is twice as long as the usual wavelengths used. So one thing is very much clear, that here in two photon microscopy, it's the longer wavelength is used, not shorter. And because longer wavelengths are used, they scatter less, they allow deeper penetration in the tissue, and the excitation is restricted to a point, and it causes less photo bleaching, which is being asked in the question. So that means option, like statement A should be correct, right? And B will be incorrect. Then if you see statement C now, statement C is the statement C is that intensity of fluorescence emitted by the sample will remain the same even if only one, okay, the keyword here is one of the two existing photons impinge on the sample. However, statement D says that no fluorescence is detected unless two exciting photons simultaneously impinge on the sample. So again, for this you should understand, to answer this question you should understand like how the wavelengths, like what wavelengths, what is the range of wavelength provided, whether it's a one or two which are provided simultaneously, right? So in epifluorescence or confocal microscopy, only a single photon of light at a specific wavelength excites a fluorophore to emit light. However, in two photon microscopy, a fluorophore can be excited by effectively absorbing two photons simultaneously. So this means that statement C is incorrect and D is correct. And therefore, the answer here will be option 2. That is A and D will be the answer, because correct inference, correct statement we have to look for. Now coming to the last question. Now this question is from subunit 13G, electrophysiological methods. In this particular question, like, or I should say, this particular question is based on EEG, electroencephalography. So if you have studied about EEG, if you know about the principle, if you know the theory, you can answer this question. So again, it's a direct question, memory-based question. So different waves of EEG are listed with their frequency. So these are the EEG waves: alpha, beta, theta, delta, and column B, the frequencies are given. Choose the option with all correct matches of the wave with its frequency. So basically, if you study about EEG, so the EEG waves, they are named based on their frequency range using the Greek numerals. So most commonly studied waveforms, they include delta, theta, alpha, sigma, and beta, right? And you can see the frequencies I have mentioned here. So basically, alpha. So from the theory, we know it should be 8 to 12 hertz. So 8 to 12 hertz means that A should matches with 3. So let's see in the options where A and 3 combination is given, only in option 3. So just on the basis of simple one wave, we can answer this question, right? But it's better to confirm. So let's take beta. So according to the theory, 13 to 30 hertz for beta. So B fourth. So yes, it is mentioned here. Then if we see the theta, it should be 4 to 7 hertz. So yes, C1 is given. And then when we talk about the delta, it is 0.5 to 4 hertz. That is, we can say less than 4 hertz. So yes, D2 is given here in the option. So this means that the correct answer is option 3. You can see that some questions were very direct. The questions asked in yeah, from unit 13, they were very direct, memory based. But yes, I'll agree that some questions required conceptual clarity. So do tell us in the comment box, in the chat box, how was your unit 13, like how you did the questions of unit 13 in the exam. And if you have any doubt regarding the unit 13, you can always contact us. We are always there to help you. So this is for now. See you in the next session. Thank you and take care.